Hello guys, welcome to Dr. Science. So today we are going to talk about the different types of translocation. In this topic I'll be mainly talking about what are meant by deletions, what is meant by balanced translocation, what is meant by Robertsonian translocation, what are meant by ring chromosomes, and what are the different types of isochromosomes. Okay, so here is a very complex diagram taken from your textbook, so I will just explain to you guys. Okay, so first we will talk about deletions. So deletion is again divided into two types, which is terminal deletions and the second one is interstitial deletions. So what is meant by the interstitial deletions? Suppose this is the chromosome here. In this chromosome, this part is lost. Okay, as you can see here, let me draw it clearly so you can understand better. So this is the part of the chromosome and this is the short arm and this is the long arm. So as you can see here, suppose if there are two breakpoints, one breakpoint is here and second breakpoint is here. So there are two breakpoints, okay, and eventually all this part is lost. Now this is called as a interstitial deletion. And what will happen to this chromosome? This chromosome will eventually bind. Suppose here was the breakpoint, so here is the other breakpoint. So two breakpoints again will fuse with each other, but here the genetic material is lost. Okay, so that was deletion. That was about interstitial deletion. Next we will talk about the terminal deletion. What will happen in terminal deletion? There is a single breakpoint and this part is deleted. Okay, these are called as the terminal deletions. So that was about deletions. Next we will talk about inversions. Before going into that, I will just talk about what is meant by ring chromosome. A ring chromosome is a unique type of deletion. Okay, unique type of deletion. So what is happening in this thing? Normally, suppose this is the chromosome present here. These terminal ends, normally these are the terminal ends of the chromosome. These terminal ends are deleted. Okay, so once after this deletion, what will happen? So this chromosome terminal end will fuse with each other. So as you can see here, they are getting fused with each other. Now they are looking like a ring, right? Hence they are called as ring chromosome. So what is happening in ring chromosome? The terminal end of the chromosome is getting deleted, both the terminal end of the short arm and the long arm. Okay, that's why once they are deleted, the terminal ends they will fuse with each other. So hence they form a ring chromosome. Okay, next we will talk about isochromosomes. What is meant by this isochromosome? Suppose this is the chromosome and this is your short arm, right? And here is your another chromosome. Wait a second here. This is the paternal chromosome and this is the maternal chromosome. So what is happening here? This is the short arm, okay, and this is the long arm. Suppose if the long arm is deleted entirely and eventually the short arms are, these two short arms are getting duplicated. Okay, then they are called as a isochromosomes. eventually you will be forming a chromosome like this. Both these are short arms, not long arms. Normally a chromosome contains a short arm and a long arm, but here the case is entirely different, hence they are called as isochromosomes. Next we will talk about balanced translocation. Okay, in balanced translocation you will be mainly talking about two things. One is balanced reciprocal translocation, and the other thing is Robertsonian translocation. So what is happening in balanced reciprocal? First we will talk about balanced reciprocal translocation. So what is happening here? Suppose this is the chromosome and this is the long arm, I mean the short arm, and this is the long arm. Okay, and here we will take another chromosome, and here is the short arm, and here is the long arm. So in balanced reciprocal translocation, what is happening? Suppose here is the length of 1 meter, and here also there is a break point with 1 meter length. So what is happening here? This 1 meter length, and this I will draw it with a different color here. This 1 meter length, they will exchange this thing. Okay, so there is a balanced, it means nearly, hypothetically speaking, we are taking 1 meter. Okay, and this 1 meter is exchanged between two chromosomes, hence it is reciprocal. So what will happen to the new chromosome? Here is your short arm, here is your long arm, so there is genetic exchange here. Okay, so this part is genetically exchanged, and here is another chromosome, and here also it is genetically exchanged with blue. Suppose this is chromosome number 1 and this is chromosome number 2. So as you can see here, genetic exchange is happening from chromosome number 1 to 2. The blue material is exchanged with the pink one, and the pink one is exchanged with the blue one. This is called as a balanced reciprocal translocation. Okay, next we will talk about the Robertsonian translocation. Robertsonian translocation. What is meant by this Robertsonian translocation? Suppose again we will take the chromosome here, and here is another chromosome. We will draw this one with a different color so you will understand. Suppose this is chromosome number 1 with pink thing, and here is another chromosome with yellow short arm. This is chromosome number 2. Okay, so what is happening here? So these are called the acral ends. These ends are called as the acral ends. So what is happening in Robertsonian translocation? The acral ends of two chromosomes will combine with each other. So what is meant by this? So this acral ends, this pink one and this yellow one, they will fuse with each other. As you can see here, this pink one and the yellow one will fuse with each other, and the long arms will fuse with each other, and eventually this is lost. Okay, and these long arms will suffice. Okay, hence this is called as a Robertsonian translocation, and this is normally seen in Down syndrome. Okay, next we will talk about inversions. What is meant by an inversion? Suppose this is the chromosome, this is the short arm, and here is the long arm, and this long arm is having 2 points. One is the pink one and one is the yellow one. So what is happening in this inversion? Yes, in this inversion what is happening is, as the name suggests, there is inversion. The pink will go up, the pink will go down, and yellow will come here. It means the genetic material is going up and down. Okay, hence this is called as inversion, and they are called as paracentric. Why they are called as paracentric, I will just explain it to you guys. Okay, now we will talk about another type of inversion. So as you can see here, here is the pink one, this is the pink area, and here is your yellow area with genetic material in it. Okay, so what is happening here? So as you can see here, here is another here is the same chromosome with exchange of material, but in this case there is a these are called as the centromeres. Okay, the central thing is called as the centromeres. So normally here there is yellow thing, and here is the pink thing. So what is happening here? Here the centromere is getting flipped, right? Or this entire thing is flipped, so we can call it as inversion. But the case here is the centromere is also getting involved, hence it is called as a pericentric. Okay, here it was paracentric because it was not involving this centromere, that's why it is paracentric. But here, as you can see, this entire thing is getting flipped upside down, right, including the centromere. So hence it is called as pericentric inversion. So now we will take a look at the diagram. So as you can see here, translocations are divided into balanced translocation. As you can see here, the blue part, this is the blue part, and this is the pink part, and there is a balanced translocation. The blue part is getting fused with the pink, and the pink is getting fused with the blue. Next we will talk about Robertsonian translocation. I already told you that these are the acral ends, these small things here. So the acral ends are getting fused, right, and they are lost. But as you can see, the long arms are getting fused with each other. Next we will talk about isochromes. What are isochromes? As I already told you, in isochromes either the short arm is lost or the long arm is lost. If long arm is lost, they will form short arms, a copy of short arms. If the short arms are lost, they will form a copy of long arms, right? Next we will talk about deletions. Deletions are mainly two types. As you can see here, there is a two break points happening here, and that genetic material is lost into fragments. Okay, next we will talk about inversion. As you can see, in inversion what is happening, there is a blue thing here and a red thing here, right? Let me zoom it. So this is the blue thing and this is the red thing. So it is going up, as you can see here, it is going up and down, so it is inverted. But the acral end is not getting the centromere is not getting involved here, hence it is called as a paracentric. Whereas you can see here is the yellow thing, here is the centromere, here is the red genetic material. So what is happening here? Red is coming here, centromere is flipped, yellow is coming here, hence the centromere is involved here. It is called as a pericentric. And you can see the colors also here. This entire thing, centromere is the important thing in the pericentric, because here in the paracentric thing there is no involvement of centromere. Okay, in ring chromosomes what is happening? There is a break point or terminal deletion, and this terminal deletion will usually form the fragments, and these terminal ends will fuse with each other and form a ring, hence they are called as ring chromosomes. Thank you.